Hello everyone, welcome to my YouTube channel. I'm going to be talking to us about something that I've been thinking, and I'm not sure if I've ever discussed that on my YouTube channel. So I feel like maybe it will benefit you guys, especially when you go for interview. You never know questions that they may ask you. Sometimes sometimes what you answer can lead to other questions that perhaps the panel they don't even have on their outline, but because of the way you answered question, it can lead to that that additional question that you may attract to yourself. I have been thinking about whether we have considered the type of blood group cells, because we know that the antibody panel cells or even the antibody screening cells, that these are red blood cells. So I've been thinking, do we actually consider the type of blood group that is used in preparation of either antibody screening cells or antibody panel cells? Okay, but anyway, before we continue, my name is Dr. Emmanuel Loto. I'm a biomedical scientist, and I'm also a lecturer in biomedical science here in the United Kingdom. So let's get into this. Now, as my custom is always, I like to, because I believe that as a scientist, beyond your answer, beyond following standard operating procedure, you've got to ask yourself, what exactly am I doing? Okay. So now the question I have here is this. We know that in blood transfusion, the routine test is blood group and antibody screening. We call it generally group and save. Okay. So now what that means is this. We need to do blood grouping and we need to do antibody screening. Okay. Now, in that antibody screening, we are using red blood cells. That's what we call it, antibody screening cells. So we're using red blood cells. Okay. And again, in antibody panels, we also have antibody panel cells. Okay. So I had already have a video on that, but I want to discuss something a bit different. So when you look at maybe antibody screening cells, depending on the type of analyzer they use, something like AutoVision, you may have something like cell 1, cell 2, cell 3. That means antibody screening cell 1, cell 2, cell 3. Or something like Bio-Rad, okay? You may have, that means IH1000. You may have something like 2 antibody screening cells. That means there is cell 1 and cell 2. Like in AutoVision, in antibody panel, you are going to have up to like, I think 12 cells, okay? But in Bio-Rad, you are going to have, about 10 cells. Let me repeat what I said before. So when it comes to antibody screening cells, under AutoVision, you are going to see something like maybe cell 1, cell 2, cell 3. That means there'll be screening cell 1, cell 2, cell 3. While in Bio-Rad IH1000, you may get something like cell 1 and cell 2. And I'm saying that in antibody panel as well, especially when it comes to AutoVision, you are going to have up to cell 12. That means cell 1, 2, 3, up to 12. But when you come to Bio-Rad, you're going to have maybe something like cell 1 to 10. But it doesn't matter the number. I just want to, bring these differences across to us. So when you have cell 1, cell 2, cell 3, or you have antibody panel, whether cell 1 up to 10 or 12, it doesn't matter. But they are cells. So these are red blood cells. So, and the question is, if we know that they are red blood cells, we need to ask ourselves, so we have blood group A, that means red blood cell A, red blood cell B, red blood cell AB, red blood cell O, okay? Now we have blood group O, A, B, and B, okay? So now what it means is that if we are having antibody screening cells and we are having antibody panel cells, so what blood group are they using? Have you asked yourself such a question? What blood group are they using to prepare those cells? Because we already know that when we are doing antibody screening or antibody panel, remember that these are indirect antiglobulin tests, or what we call indirect cum tests. In other words, what we are measuring is not on the surface of the red blood cell as it were, as per the patient, okay? What you are measuring is actually in the patient plasma. But we are going to use something that is on the surface of the cells, okay? We are using what is on the surface of the cells to determine what is in the patient plasma. Does that make sense? Okay. Now, if you understood that, we now need to ask ourselves, knowing fully well that when we say blood group A, it has antigen A, blood group B, it has antigen B, blood group AB, it has antigen A and B, blood group O, we say it doesn't have antigen A, it doesn't have antigen B. But that is in terms of ABO grouping. I had already said many times on my videos that when you talk about these blood groups, we have IgG antigens. Okay, so when you say blood group A, when you say antigen A or antigen B or AB, as the case may be, okay, even when you say antibody A, antibody B, okay, what you are referring to is IgM, whether antigen or antibody. Okay, now I had said that when it comes to IgG, every red blood cells has IgG antigens. Whether it is O, whether it is A, whether it is B, whether it is AB, they all have different IgG antigens. And I had already said that in this IgG antigen is not respecter of a particular blood group. What that means is that you may see something like antigen K on blood group O, you can see it on A, you can see it on AB, you can see it on B, as the case may be. So you can see any IgG antigens in any blood group. At the same time, you may not see any IgG antigens in any blood group as well. In other words, when you say blood group O, you may see antigen K, you may not see antigen K. So it varies depending on the DNA, the coded DNA gene of that very individual. Now, back to what I want to answer. What if they are preparing antibody screening cells, which are red blood cells? If they are preparing antibody panel cells, which are also red blood cells, which blood group do you think that they are using? Now, let me go straight to the answer. So when they are preparing that, because it is a cell, remember, we need to make use of that cell that does not have the IgM antigen. So remember that blood group A has antigen A. So if you use blood group A to prepare antibody screening cell or antibody panel cells, what it means is that when you use the patient plasma, remember you're using the patient plasma. So the patient plasma, if it has antibody A, it will bind to that antigen A. And that can give you a false result. Because when we are measuring IgM antigens and antibodies, what we want to measure is ABO grouping. When we are determining forward or reverse grouping, we can make use of this IgM. But when it comes to IgG antigens, our focus is on our focus will then be on not on this IgM. Our focus will be on IgG. I mean that when we want to do antibody screening or we want to do antibody panel, our focus is no longer on IgM. Our focus is now on IgG. So what it means that if they are preparing antibody screening cells or antibody panel cells and they use blood group A, that blood group A has antigen A. And what it means is that if you try to do antibody panel or you try to do antibody screening of a patient that is blood group B that has antibody A, that antibody A will react with that antigen A of that cell of blood group A. That means blood group A, we can't use it. Now let's come to blood group B. Blood group B has antigen B. If you use blood group B to prepare your antibody screening cells or to prepare antibody panel cells, what it means is that if you do antibody screening or antibody panel of someone that is blood group A that has antibody B, that antibody B will then react with that antigen B on the surface of that very cell. Are you following me? That means blood group B then is not recommended. Now, obviously, because of that, when you use blood group A and B, blood group AB, so this blood group AB has antigen A, it has antigen B. It even makes it even worse because it means that when you use blood group AB to prepare your antibody screening cells or antibody panel cells, what it means is that when you do the antibody screening or you do the antibody panel, if the person is blood group A, that antibody B of the blood group A will react to the antigen B that is in that blood group A and B. If you do antibody panel or antibody screening of someone that is blood group B that has antibody A, that antibody A will react with that antigen A. Does that make sense? Because it has the antigen. Okay. Now, because of that, it becomes complicated. That is why whether you say blood group A, whether you say blood group B, whether you say blood group AB, you can use them to prepare antibody screening cells, and you cannot use them to prepare antibody panel cells. The reason is because they have this antigen A and antigen B, or both antigens. And if that is the case, if you use them to prepare it, any patient that has any of the antibody that is specific to any of the antigens is going to bind, and that will give us a false positive result. And that is why when they are preparing, whether it is antibody screening cell or antibody panel cells, they are going to use blood group O. The reason why they are using blood group O is because blood group O does not have this IgM antigen A, does not have this IgM antigen B. So because it doesn't have IgM antigen A, it doesn't have IgM antigen B, what it then means is that whether it is blood group A, whether it is blood group AB, whether it is blood group B, whether it is even blood group O, it doesn't matter. Even if you have antibody A or antibody B or antibody A and B on the patient plasma, because there is no antigen A, there is no antigen B on the blood group cell, it cannot bind to it. Does that make sense? So that is why we use blood group O to prepare our antibody screening cell and antibody panel cells. I hope that makes sense. Now, the reason we are doing that is because our focus is not IgM. Our focus is IgG. What it then means is that if there is then any positive reaction, whether in antibody screening cells or whether it is in the antibody panel, so once you see that there is positive, there's a reaction, you know it cannot be antibody A, you know it cannot be antibody B. Why? Because you know that that cell does not have antigen A, it does not have antigen B. So that is why when we see the positive on the antibody screening cells or positive on the antibody panel cells, we now begin to suspect the IgG antigens. Because I have told you that these blood groups, irrespective of their different blood group, now whether it is A or B or on this occasion, now we're using blood group O. So blood group O has different antigens, like other blood group in terms of IgG, by the way. Okay, in terms of IgG antigens, I have told you you can see them in any blood groups. Okay. And these IgG antigens, there is no way IgM antibody A or antibody B will bind to them because they are not specific to it. Does that make sense? So this blood group O that is used has different IgG antigens, whether it is a D antigen, whether it is C antigen, K antigen, M antigen, N antigen, whatever, any of the IgG antigens can be expressed in the blood group O. So what they do then, they now sample these blood groups O and use them to prepare these very these very antibody screening cells, and also use them to prepare these very antibody panel cells. What it means is that when you say cell 1, cell 2, cell 3, cell 4, so it is from there. These are individuals. Are you getting me? These are different O cells that are specific to different antigens, and that is why they can work out having sample 10 cells, okay, or having sample 12 cells, okay, if it is antibody screening cells, having sample 1 cells or 2 cells or 3 cells, they have sampled the cells. They know, okay, this particular cell is positive in here, 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 but not positive here, here, here. So they will now use up to that 10 cell to work out, that antibody panel you are identifying, or the antibody screening that you're using to know the possible antibody that may be present. So the final answer to that, like I've tried to say, is the fact that we use blood group O to prepare our antibody screening cells and our antibody panel cells. And I hope that makes sense, because I have said that the blood group O does not have antigen A, does not have antigen B, therefore cannot react with any antibody A or B in the patient plasma when you're doing your IAT. Okay, when you're doing your IAT, that means Indirect Antiglobulin Test. Okay, however, this blood group O has IgG antigens. So they will sample different blood group O antigens and see where they are positive and use them to prepare both your antibody screening cells and your antibody panel cells. So that is the answer to that. So if you go for interview, they ask you, can you tell us the blood group, the type of the blood group used to prepare antibody screening cell or antibodies, identification cells? That is blood group O. And if they ask you why, you say because blood group O does not have antigen A and does not have antigen B, okay? And therefore cannot react, therefore any antibody A or antibody B in the patient plasma cannot react with them. And that helps us to know that any reaction we are getting is not due to IgM antibodies, rather it is due to IgG antibodies, okay? Now, remember, we are using cell, which is an antigen, to identify the antibody, okay? So, because it's important that you put your terminology right. So, the cells, whether antibody screening cells or antibody panel cells, these are antigens. But in the plasma of the patient are the antibody. So, we're using these antigens on the surface of those cells to identify the antibody in the plasma. So, those cells refer to antigens. The patient sample refer to what? Antibody. So when antibody screening is positive or when antibody panel is positive, what it means is that the patient sample, plasma or serum, is specific to a particular antigen. That means the antibody in the patient plasma or serum is specific, bind to a specific antigens on that cell. So that way you can say, okay, this person has that antibody. Okay, all right. And like I've said so many times, anytime you identify antibody, it invariably means that the person does not have the antigen. And that's why you select the antigen negative to do cross-match for that patient. All right, thank you so much. I hope that makes sense. Please do let me know what you think about the video. Can I ask you to please like, share, subscribe? Make sure you subscribe, okay? Make sure you subscribe and share with your friends and family. And I wish you all the best till I come back your way again. Bye-bye.